All right. Thank you all for today joining the webinar. We'll be talking about scientific agents for biotechnology R&D. I'm Berke Buyukkucak. I'm the CEO and co-founder. We're also joined by some of the team. One of them will be presenting as well, Eva Alderman, who's our product manager, who can introduce yourself in a bit. But today we'll focus in on, as I mentioned in the email, the pipeline mode. It's essentially a new way that we've developed using sort of agents to link up different types of AI models for different types of use cases. We're excited to show you essentially how this currently works. It's sort of how it moves us closer to real agentic science. And if you're interested in collaborating, you can note my email down, Berke@superbio.ai, and feel free to shoot me an email. This email will also be at the end of the slides. Moving on just briefly about the agenda. Quickly, we'll just want to talk about sort of the motivation, why we're building these scientific agents, why we built the pipeline mode, and essentially the problem it solves. Next, we want to talk about the use cases, right? What type of use cases, especially in the early drug discovery R&D, our platform currently is addressing the best, and what type of tools biotechs are using today. Then we'll move into agent evaluations, right? We're going to put our system head to head with sort of LLMs like GPT-4, GPT-03, and see how they compare. And finally, we'll wrap up with a Q&A session. We'll answer. Some of the attendees already shared some questions with us, so we were really excited to see them. So thank you for that. So we'll be answering those questions and getting questions from the from you all as well. So before we start, I'll just to set up a bit of a context, why we're building agents, I'd like to demo the Superbio App Store. When we first started a few years ago, we essentially vowed to become the de facto AI store for biology. So what we've done is we collected hundreds of AI models for biology use cases. We deployed them on our platform, and we sort of painstakingly built very easy to use interfaces over every single one of them. So this is the Superbio App Store. Each card here represents a single model. At the top, you have the name, the author, the organization it comes from. You have a very short explanation of what it does, its category, its usage stats, how many times it's been used by the community, how many times it's been liked or favorited. And if you're interested in running one of these models, all you need to do is just click on the app card. That takes you to the app page. We have different categories around protein design, omics, image analyses, and again, all of the models, their interfaces look pretty similar to each other. This is pretty standardized. If you'd like to use it with your own data, you can upload your own for RF Diffusion. This model that comes from Baker Lab, you can upload your own PDB file from your computer, from the Data Hub, our built-in data management system, or different remote sources, right? And if you just want to test it, we provide you demo data. And the next step 2, is setting the parameters. Here, either you can sort of read about how to set these. We also provide helpful tooltips on how to set these parameters. Once you're done, all you need to do is maybe set up a job name, that's optional, or run this on the Superbio infrastructure, right? The results, essentially, it completes in a few minutes. They come back in sort of a more structured, reproducible format. You have the explanation of what the results are. You can view the preview here. You can also download these output files to your local, right? So the App Store, essentially, RF Diffusion then with other hundreds of models is the foundation of our platform. We collected all these models, deployed them. Not only you can use them through the user interface as a scientist, but also as APIs, right? So pipeline mode really builds on top of this by being able to chain multiple different models to solve different types of use cases, right? And just to talk about the motivation, right? Why we went down the agentic route. One of the biggest things that we heard from users that's been interacting with the Superbio App Store is, one, I don't know which AI tools to use, right? I come to your platform, they have all these great names, RF Diffusion, AlphaFold, but I don't know which one's the best for my use case, and I don't know how to use them, right? I don't know, even though you built a nice looking interface, I still need to set the parameters, right? So that has really been a challenge. Secondly, again, to design a protein binder, for example, just using one of these models usually is not enough. You need to use a chain of models. And usually when biotechnology companies, pharma organizations come to our platform, they just say, hi, I want to build a one-click pipeline. I don't want to go into every individual model, run it myself, download the data, re-upload to the next one. I just want you to be able to build it for me, right? And finally, they mentioned that they want to customize models, right? They, take an existing model, fine tune it with their own data, or to be able to, change the parameters a little bit. All of these, of which we found could be quite doable with agentic large language models that can actually understand queries and execute on them later on, right? So this essentially showed us a need for scientific assistance to help both navigate the Superbio platform as well as the larger ecosystem of AI tools that's out there. Finally, just a brief note about the use cases. Today, and this is the final slide before I actually pass it to Eva, and you can see the pipeline mode in action. Just move my cat real quickly from in front of my screen. The platform today, the pipeline mode really excels at a few different use cases. One of them is protein design, like things like de novo binders, VHHs, as well as like a ligand-aware protein design. Secondly, it's really good at structural biology. There's a lot of AI research that's going into this. We can fold proteins. We can analyze full biomolecular assemblies like protein ligand, in some cases protein iron, right? And then we have a good sort of computational chemistry tool that allows you to design sort of de novo drugs, molecular docking, as well as make affinity predictions with perhaps a model that you've all heard with Bolt2, right? On the right, you see sort of a pipeline that's been generated by our pipeline mode, right? It chains together this quite well-known models of diffusion protein NP and AlphaFold 2 to be able to give users a pipeline to generate protein binders, right? The key point here is that the models that we put together in our platform in the Superbio App Store are not really isolated tools anymore. We built out this pipeline node that can connect end to end and allow users to create these complex architectures. So without further ado, I'll pass it to Eva to show us the platform in action.
Awesome. Thanks so much, Berke. Can everyone hear me okay? Okay, wonderful. So as a way of quick introduction, my name's Eva. I'm the product manager at Superbio, and I actually hail from a bench background myself. So I got my start in CAR T discovery and development before getting interested in informatics and moving over to immunogenomics from there. I think, in the last couple of years, I've been very captivated, as I'm sure everyone else has, the capabilities of large language models and what they can do in science. So that's what I've been working on here with Berke for the past year and a half, and I'm excited to show you some of our progress there today. So I will start sharing my screen. And our most recent product release is this pipeline builder, which actually uses a series of scientific agents to first select tools on the Superbio platform in response to a user query, and then build a workflow and actually help you set the parameters for that workflow. So the end-to-end process is about 10 minutes total, typically. And so that's not totally practical to share with you today over a webinar. However, we will first share this sped up video. I'll then show you some of the workflow outputs that have been generated in platform, and then we'll end by actually showing you how to watch one and hoping that you do so and give us some feedback. So I'll start playing this video that I've recorded here. This is a demo of a de novo binder design task, focusing on generating a de novo binder hypothetical task targeting the SARS-CoV-2 RBD domain. So the first thing that happens when you enter a query into Superbio is that a series of follow-up questions are generated. So this is a bit similar to the deep research product from OpenAI, if any of you have tried that. It then uses that information to actually generate an improved prompt that can offer more information to the system. So you can accept the prompt, you can edit it, you can decline it, it's totally up to you. After that, the system starts searching through Superbio's App Store and actually, as you can see here, suggests a series of applications that could accomplish the task. One thing you can do is you can actually ask questions of the system at any time. So in this case, I'm showing perhaps the user has a question of why did you choose Protein X for this pipeline? And the system can offer an explanation. If you like, you can also suggest tool changes at this time or any other edits to the workflow. The system will then search literature and web search to actually derive potential parameters based on sort of existing biological and technical knowledge and offer a completed workflow as you see here. So now that you've seen that, I'd like to actually show you just in platform what some of these workflows look like. So here I'm showing the generated protocol that we just demoed. I'm showing the chat over here of the interaction that the user had with the system. So we've kept this. You might notice if you've used vibe coding products, the UI is somewhat similar. It was important to us to have this chat interface where the user could ask questions of the system and sort of provide feedback. You then have a generated pipeline to your right here. We also offer sources, literature and web sources that were actually used to help set parameters in this workflow. And you can download a report over here of all of this information. So just to go through, I did actually load up some workflows for additional use cases that I wanted to show you very quickly, just to get a sense of what the platform can do. So for example, one area we're excited about is this emergent therapeutic modality, macrocyclic peptide binders. So I've shown here going through the similar process, asking the system to de novo generate a macrocyclic peptide binder against a typical surface target, CXCR4. So once again, going through a similar process for workflow generation. I just wanted to highlight over here that for the workflow parameters, like others, others developing AI systems, they do sometimes make mistakes. So we've made it possible to actually edit any of these parameters. So for example, if I wanted the peptide to be a different length than the system suggested, I could edit that parameter and save it over here. You can also peruse the reasoning steps that the agents have output, just to sort of get a sense for the justification of any of the parameters that were set, the applications that were chosen. And this is also included in the final report. Another thing that I wanted to highlight, so I've done sort of a mock protocol here, asking the system to estimate the permeability and solubility of caffeine. So this leans more into our computational chemistry use case. And the system here has suggested a series of applications that can estimate these ADMET properties. Another thing that the system can do when it searches these external sources is it can actually return, in some cases, PDB IDs for different files, or actually SMILES chemical names. So I just wanted to show here, this is usually included in either the text of the protocol or sometimes the workflow parameter, just depending on the workflow. Another thing I wanted to show is that a question that we get a lot and that we actually got ahead of time for this webinar is, how do I know that these parameters are accurate and real? So we do provide sources such that you can actually cross-check these yourself. So, find the appropriate publication, go look to see if some of them actually make sense for your scientific use case. But I just wanted to show here that we do actually have another feature rolled into this Copilot feature in platform that can help you do this. So for example, for RF diffusion, it's essential to choose a target region on the target protein that actually makes sense for the use case at hand. So in this case, generating a binder targeting the SARS-CoV-2 RBD domain, we're really looking to disrupt that RBD human ACE2 interaction in the therapeutic context. So one thing I could do is I could sort of copy and paste the hotspot input here, which refers to key residues whose interaction we'd like to disrupt on the RBD surface. And I can actually go to our search Copilot, which is another feature rolled into here. And I can ask, do the following hotspot residues actually make sense? Are they scientifically viable to use for this particular use case in this particular workflow? And the search Copilot, which I'll also show you how to launch from our interface, actually searches with more breadth and depth the available existing literature knowledge, and can actually return here numerous citations and explanations of the information in the citations, as well as a review table. So it actually goes through every single residue and sort of tells you a bit about the role in the binding. So I think at this point, Berke, we should, I will actually finish this demo by showing you how to actually launch a workflow in the Superbio Copilot. So first, in order to do the workflow generation, you click here on Build Pipeline. You can then see a dropdown of the various use cases, so protein design, structural biology, and computational chemistry, along with more specific use cases. So for the sake of this demo, I will say, let's fold one GFP from its sequence. I'll then click Enter, and the system will begin by thinking and then generating some follow-up questions. So typically these questions are geared at additional detail, such as, would you like to use publicly available data, or will you be bringing your own? It will ask whether you're interested in additional capabilities that Superbio can provide. or whether you'd like to provide additional scientific context for the task. So in this case, I'll say use public data. For number 2, I'll say no, thank you. And then number 3, I'll also just decline at this time. The system will then use that to generate an improved prompt. So as I mentioned before, you can either accept, decline, or actually edit the prompt at this stage. So perhaps I'll just say, or I'll actually just edit some of this away without additional, there's some extra text here that is unnecessary. So I'll then accept the prompt. And after that, the system will begin the process. So I think, Berke, this would be a good time to actually return to the slideshow. Yeah, so one thing that we want to discuss is the accuracy of the system. This was something that is both very important to us for convincing ourselves that that what we built here is worthy undertaking, and also improving the system over time. So we designed 2 sets of custom evaluations to help us evaluate the agents that we were building versus existing large language model products. So this would include what we're showing here are the sort of some of the latest frontier models from OpenAI, the O3 reasoning model, O4 mini, GPT 4.1, and GPT 4.0, and also against the Superbio agent. So the way that we design these evaluations, one thing I want to emphasize is that these are not broadly used evaluations, because the set of things that we were trying to assess were quite specific to our platform, specifically whether an agent could select the best tools available on Superbio for a query is the evaluation we're showing here. So we designed a series of 42 pipeline tasks. As you can see on the right, the example task here is asking the system to select the best computational pipeline on Superbio to address the following query. So as our example use case, the query is designing a protein binder targeting the SARS-CoV-2 RBD domain. We give it a series of multiple choice options here, which are all quite different from each other. So option A is actually a de novo small molecule drug design query. Option B is a protein design query, similar to the one that we just showed. Option C is more geared toward genotype-phenotype relationships, and option D is a foundation model for single-cell genomics. So our agent here actually uses OpenAI's O3 as the base model. However, what we've done is we've built a knowledge context based on Superbio's existing app store. And then we've also provided some external search tools that sort of help with decision making. So we do see a marginal improvement here. What we're showing is the results of 3 technical replicates to account for the sort of inherent variability in agent and LLM function. Next slide, please, Berke. We also had some questions. So we use a separate agent to actually search literature and web to help set parameters, as was shown in the demo earlier. So on the right here, you can see an example of the evaluations that we've designed for this. We had about 50 tasks here, and the evaluations were designed similarly in the sense that they were multiple choice, so that we can get a very nice binary ground truth readout. And in this case, the system was given a series of potential RF diffusion parameters for a binder targeting EGFR variant 3. So what we do see is that we do see a small, also marginal improvement here. So yes, so this is, again, the result of 3 technical replicates, giving the system a series of parameter and pipeline evaluation tasks. All right, any more questions about that? Or next slide, Berke. So we do want to note that, I mean, as you can see, even in the format of multiple choice answers or evaluation tasks that we provided to our agents, they do still make mistakes. In the open-ended context, they do still make mistakes like any AI system. And I just wanted to highlight this quote from Sundar Pichai, that the future of AI is not about replacing humans, it's about augmenting human capabilities. We think for science tasks, the cost of a mistake is quite high. I mean, for example, the incorrect parameters for an algorithm like RF diffusion could cost significant wasted time and resources in the lab. And that is why we've endeavored to keep the human in the loop every step of the way, so that scientists have full control over what the system is doing. So, all right, yeah, next slide, Berke. I think we might be ready to move on to our Q&A section. So I think I'll start just with FAQs. Perhaps I can take number 1, Berke, and then pass it to you, and I can sort of review questions as they come in. Number 1, how does your platform propose to combat misinformation from hallucinations? I would say the number 1 thing that we've done here, as many of you who are familiar with the frontier reasoning models may know, they do have quite high hallucination rates. when they are not provided with retrieval augmented generation tools or other search tools. And we do find that providing them with these tools, as we've done, so this would include our literature search tools and also the knowledge context we've created for Superbio and given the ability to search, we do find that the hallucination rate comes down significantly. However, in our internal evaluations, this was still sitting at about 5%. And indeed, hallucinations do happen. So that is, again, the sort of product rationale for keeping the human in the loop here.
Thank you for joining us, everyone. It's been great. I know we are a few minutes over time, but hope this has been useful. Certainly your questions have been quite useful for us. Thanks so much. Have a wonderful day.